Hello friends, welcome to BMH Learning. In the last part of this lecture series, we talked about the activation of the receptor tyrosine kinases. In this video, we are going to discuss about the JAK-STAT pathway. I recommend you to go through the first part because this video is totally based on the first part. So there are two types of receptors which activate the tyrosine kinases. The first one, in which the enzyme is an intrinsic part of the receptor. The second one, in which the enzyme is not an intrinsic part. In this case, both the enzyme as well as the receptor, they are encoded by different genes. This JAK kinase is an example of the second type, okay, where both the genes, where both the receptor as well as the JAK kinase, here the enzyme, okay, they are encoded by different genes. Although they are different, but their activation process, which is starting from the ligand binding to the receptor and then activation of the associated enzyme, is same. So when the enzyme, here the JAK kinase, is activated, what happens next? This enzyme would phosphorylate the tyrosine residues which are present on the cytosolic domain of the receptor. Now what is the purpose of these phosphorylated tyrosine residues? They bind to the proteins which are present in the cytosol. So there are proteins which are present in the cytosol or the cytoplasm in inactive form. Those inactive proteins, they come and bind to these phosphorylated tyrosine residues on the cytosolic domain of the receptor. And when they are here, they are acted upon by this JAK kinase. And after its action, those proteins become activated. And what is the function of those proteins? They act as transcriptional factors and help in the transcription of genes. One such protein which is activated by JAK kinase and is present in the cytosol as in inactive form is known as STAT. And on this STAT, this pathway is named as JAK-STAT pathway. So what is the full form of this STAT? This is signal transducer and transcriptional factor. So as the name suggests, this STAT protein serves two functions. The first one is signal transducer. Okay, the signal is coming in the form of ligand, right? From ligand, the signal is transferred to receptor. Okay, from receptor it is coming down and finally to this STAT protein. From STAT protein, it would go down into the intracellular targets. The second component we have is the transcriptional factor. In molecular biology or cell biology, a transcriptional factor is a protein which is useful in regulating the activity or the function of the gene by increasing or decreasing the rate of the transcription. Okay, so before we discuss the mechanism of this JAK-STAT pathway, we need to know the components of this STAT protein. So we have three components. The first one is the DNA binding domain, this one in blue color. For the transcription of the genes, this STAT protein, it must bind to the DNA. Okay, we know the protein has two terminals, the N terminals and the C terminals. This DNA binding domain is present on the N terminal. So this is the N terminal DNA binding domain through which the STAT proteins bind to the DNA. The second component we have is the SH2 domain, which is the most important. Okay, for the activation of this STAT protein so that it can initiate the transcription of the genes, there must be a connection between this STAT protein and the this receptor tyrosine kinase, in this case JAK-STAT. Okay, so there must be a connection between this STAT protein and this receptor. This connection is made between one domain of the STAT which is known as SH2 and here one of the tyrosine residue in phosphorylated form which is present on the cytosolic domain of the receptor. Okay, the third component we have here is the domain with the tyrosine residue. This is present on the C terminal end of the STAT protein. Now, the activation of the STAT protein is not done by the binding of the SH2 domain to one of the phosphotyrosine residue which is present on the cytosolic domain of this receptor. Actually, when this SH2 domain binds here, so this STAT protein is here, right? Then we have a specific tyrosine residue on one of the domain of this STAT protein present on the C terminal end of the protein. This tyrosine residue is phosphorylated by JAK kinase, and then we get the activated protein. Now we will discuss through this diagram how it goes. So initially the STAT protein is found in the cytoplasm as in inactive form, and when the JAK kinase phosphorylates some of the tyrosine residues present on the cytosolic domain of the receptor, it sends this phosphorylation, this inactive protein, and comes here and bind to one of the phosphorylated tyrosine residue through SH2 domain. And when this happens, the JAK kinase, okay, comes into play, this one, from here through its activation lip, it phosphorylate on the domain with the tyrosine residue. There is a specific tyrosine residue here. This one is phosphorylated by JAK kinase. You can see here, this one is phosphorylated, and this phosphorylation makes this protein, the STAT proteins, active. Now, what is the purpose of this activated proteins? Of course, we know this is a transcriptional factor and will go into the nucleus and activate the transcription, okay, or initiate the transcription. But before it go into nucleus, there is a condition. Okay, it cannot directly slip into the nucleus. It must go through one more step, and that is known as dimerization. As a monomer, this protein cannot go into the nucleus. It needs to dimerize. Now, why there is a requirement for the dimerization of these STAT proteins? So there are two reasons for that. In this video I will tell you one, and you have to find the second reason yourself. You can comment in the comment section. For this protein to move into the nucleus, it must have an attached address, so that it would only go into the nucleus and not somewhere else. That address is in the form of nuclear localization signal. So there is a nuclear localization signal which is only found in the dimeric form. When these STAT proteins, they are present in monomers, this NLS is covered, is hidden. When they form dimer, this NLS is exposed. Okay, when this NLS is exposed and it is here, it would escort this dimer of these proteins, which is here, okay, into the nucleus. And when this is in nucleus, along with other proteins, other transcription machinery like coactivators and activators, and of course the RNA polymerase, it would initiate the transcription of the genes. I hope this video would be helpful for you. Thanks for watching.